Perfect.
Can you see my screen now?
Yes, we can, and now we are recording. So whenever, so just as a reminder, these talks are informal and it's just a conversation about our research, key concepts, methods, and results. It doesn't have to be the talk that you're gonna give in the symposium, but it's rather just an overview so that you can get feedback that could help you build that talk. So thank you, Carmen, for being the first one. I'm gonna ask everyone to mute themselves unless they have any comment. They can, of course, unmute themselves, but so that the recording is good quality, we will mute ourselves except Carmen, that will have her microphone on.
Okay. Thank you, Martha. Thank you very much. Thank you very much, everyone, for being here today. I'm not sure if I should be the first one. I'm very nervous today, so please, let's try to do very informal and ask me any question you have during my presentation. And Marta, if you think that there is something that you can express better than me in English, please stop me and let me know, okay? Okay, so I don't know if I can see all of you. Ron, I lost your Kitties. Where is your Kitties? Your Kitties is the name?
I don't see your case anymore. I think that maybe they are not in the meeting anymore, but they will probably come back if they are available.
Okay, so I think that first of all, I'd like to introduce my colleagues that are today here. Antonio Ibáñez, Antonio David Rodríguez, Alejandro must be over here. Alejandro is here. So these are my colleagues. They are members of my research team, and they are the ones that are helping me to develop this project that I plan to present in SFN, hopefully, if we have good data, I hope so. So Alejandro and Antonio are working with the CFOS expression analysis and with the immunohistochemistry, and Antonio Ibáñez is our expert in the graph theory analysis. So I was reading about the graph theory. So I hope you don't ask me any difficult question about the graph theory analysis. So this is just an initial approach of the thing that we want to do. So I hope we can do it soon when we have enough data to start with this mathematical analysis, okay? So thank you for being here today. Thank you, Masaki. I don't know what time is in Japan now. It's very early.
No, yes, 5 a.m.
5.
It's fine.
You know, my daughter is in Tokyo right now.
Already?
Yes, yes. Yes, she's enjoying a lot. Yes.
That's good.
Okay, so let's start. So when I was thinking about the content of my presentation, I was thinking about presenting only my current research, or rather describing all my journey, research journey along the last more than 30 years. So I decided to do the second, because I think it's good if we can present ourselves to talk a little bit about our research trajectory. So in the topic of frustration research. So my journey starts and ends in the same place. So this is the city where I was born. This is Jaén. It's a province of Spain. It's located in the south of, hi, Jokiris, in the south of Spain, in Andalusia. It's very close to Granada, Cordoba, and Seville, that are very famous cities. But Jaén has millions of olive trees. Is very famous because it has a high quality olive oil. So if you like olive oil, you are invited to come to our city to visit our laboratory to enjoy our landscape and our olive oil. And my journey starts here because I was born here, and because when I started to study psychology here, this is me and the University of Jaén in 1985. I was like 20 or so. So this is the campus a long time ago. We only had 3 buildings, and this is where I studied my bachelor degree in psychology. And this is where I start, and this is where I end, because this is the University of Jaén today. Here we can see that we still have the same building. This one is a building for teaching. We also have this old building, and now we have this building is for research, this one and this one. And this is where the animal rats, where the rats and other animals are located for training. This is the library. So our university expanded a lot. It's still very small, but it's nice. This is my last picture in the laboratory last week, with some of my undergraduate students and also with our technician. So we are just now running an experiment, and I want to show you some data, preliminary, because we are still in the process of running this experiment, and I will show you some ideas that we have. So between the start and the end, I went to the University of Granada to develop my dissertation, my PhD, my doctorate, and I was working there and studying there for 6 years, and then under the supervision of Antonio Cándido and Antonio Maldonado from the Learning Research Group. After that, I had the opportunity of visiting a number of important universities and collaborating with people focused on frustration research too. So I spent a summer in the Dr. Flaherty's laboratory in Rutgers University. I also visited the laboratory of la doctora Alba Mustaca in Buenos Aires, Argentina. She sadly passed away last year, and this is a picture of his research team, where I spent 2 months working also on frustration research in animals. And during the last years, I have visited very regularly the laboratory of Dr. Mauricio Papini, that is not here today because he enjoys in Hawaii, in Honolulu, right now with his sons and his grandson. And this is a picture with some of his students. So I have been visiting his laboratory and learned a lot from his experience. These are some of the colleagues that have also collaborated with me. So I'm here thanks to all of them. And these are students and faculties from many different universities, not only in Spain, but also in Germany and in Buenos Aires. So thanks to all of them. These are some of the people that are currently collaborating in our grant. So this are Antonio, Tony. We have many Antonios and many Martas. Antonio David Rodríguez Agüera, that is here. Santos Blancos, that is from the Department of Biology. Alejandro Navarro, that is also over here. Marta Sabariego. Marta, you are included as a member of the department of the University of Jaén. I'm sorry, because you did your thesis here, so you are a member of our team also. Marta Valero, that is also over here, is our graduate student now, and she's conducting all the CFOS stuff. So if you have any question, you can ask her. Hi, Marta. So as all of you know, one of the topics that all of us are interested in is the frustrating non-reward effect that can be defined according to our definition. The definition we have been working on is the experimental procedure of removing, omitting, or reducing reward quality or quantity in the presence of signals or situational cues previously paired with rewards. This is the definition by Amsel in 1958. And the frustrating non-reward effect can be studied with a number of experimental manipulation or procedure, and I have been working for many years in the successive negative contrast effect. Not only in this effect, but today I'm going to focus on this effect. The successive negative contrast effects can be defined as a temporary reduction in responding to a small reward in animal previously exposed to a larger reward, in comparison to the responding observed in a control group always exposed to the small reward. This is the basic phenomenon, the basic effect, and as you can see here in the table, in order to obtain this effect, we need to use two groups. One group is the downshifted group, and the downshifted group is exposed during a preset phase to a high reward value that is suddenly and unexpectedly downshifted to a low reward value, and the performance of this group is always compared with a control group, an unshifted control group that always receive the low reward value throughout training. So the basic comparison is between the downshifted and the unshifted group in the post-seed phase. So if we see significant differences between the downshifted and the unshifted group, is because reward has a relative value and the animal is comparing the actual reward with the expectancy of the memory of the previous reward. As a consequence of this comparison, a frustrative non-reward response trigger, and this negative emotion is responsible for the response impairment that we can observe in a variety of experimental tasks. So, did you understand my explanation? Is clear? Marta?
Yes, yes, it's clear. Marta S. says that it's clear.
Marta Sabariego.
I think it's clear. Thank you.
Okay, so please ask me any question you have, okay? Okay, this is a typical result, a typical figure, where you can see the performance of the downshifted group in the preset phase, and the performance of the downshifted group also in the preset phase. We usually see differences in the preset phase because the experimental group is receiving a higher reward, and according to that, we usually see a better performance. And after the downshift, what you can see here is that the comparison shows performance impairment in the experimental group, in the downshifted. Regardless, the dependent variable that we use, we consistently see in the experimental group that after the downshift, the response goes away or the response worsens. This is usually a transient phenomenon, so we cannot see this recovery here, this complete recovery, but it's an effect that usually recovers relatively fast. So by the end of the sessions, you can see similar performance between the control and the experimental groups. Okay? So during my journey on frustration research, I use a variety of tasks, and I want to show you just a summary, a brief description of this task and some of the results that we have obtained using this task. During my doctorate, when I was working at the University of Granada, I was working with an aversive instrumental successive negative contrast task. But we have used at the University of Jaén also this task, the aversive task, and the appetitive instrumental and the consumatory successive negative contrast tasks. So let me describe a little bit this task. The first one is the aversive instrumental SNC tasks. It consists in a one-way avoidance task in which animals are exposed in an unsafe compartment or a danger compartment to a warning signal that can be a light, like here, or a tone. We used to use a tone in our laboratory. This is a warning signal that is followed about 5 seconds later for the presentation of an electric food shock. So the animal can have the opportunity of running and go from the danger compartment to the safe compartment. So if it runs from the danger to the safe, he will be able to escape from the tone and to avoid the electric food shock. That will be a correct response. So according to some theories, one-way avoidance training involves two motivational components. One of these is fear, and fear is triggered in the safe compartment and promote flight response. So the animal that is feeling fear here will tend to flight and go to the safe compartment. But in the safe compartment, there is a very interesting motivational component that is relief. Relief associated with the termination of the warning signal. So the relief can promote approach behavior. So after extensive training, the avoidance response is like a mixture between flight from fear and approach to relief. So according to some theories, signals associated with the absence of the shock, that is safety signals, can develop reinforcing properties. So if they have reinforcing properties, then we can manipulate them as appetitive rewards. So let's imagine time spent in safe compartment as an appetitive reward as food. So we can manipulate, we can increase or decrease time spent in safe compartment, and if safe compartment has reinforcing properties, we can induce a successive negative contrast effect, similar to that obtained in appetitive tasks. Okay, so this is what we did. This is the typical experimental design. Here we can see the downshifted group. This group was exposed to 30 seconds in the safe compartment in the pre-shift phase, and then was downshifted to 1 second of permanence in the safe compartment in the post-shift phase. Okay, and the performance of this group was compared with that registered by an unshifted control that was exposed to the avoidance task or to the safe compartment only during 1 second. Okay, so this is high reward. Okay, this is high reward. Can you see my mouse? Yes, right? So
Yes.
this is a high reward, this is low reward, and this is low reward, and this is low reward. And again, we compare performance of this group during the post-shift phase. Okay? The dependent variable was trials to criterion. So how many trials the animal needed to complete a number of consecutive avoidance responses. Okay, so here we can see the result of one of the experiments that we conducted. Here you can see the performance in pre-shift phase and in post-shift phase. And the most important comparison is that of the contrast group and the control group in the post-shift phase. So as you can see here, even though both groups are exposed to 1 second in safe compartment, the performance of control group is very good, but you can see an impairment in the performance of the contrast group. So the number of trials needed to reach the criteria significantly increases. So they needed more trials to reach the criteria because they were receiving the shock. They were not escaping. They stayed in the safe compartment because of the devaluation of the safe compartment. So the relief is lower because the animal can only stay 1 second in the safe compartment. So this is like a reward evaluation procedure. Okay?
Sorry, can I ask you a question?
Yes, please.
Yeah. So in this paradigm, what is the reward? Reward is the time there. How
The reward, sorry, the reward?
Yeah, what is the reward in this?
The reward is the time, sorry. The reward is the time spent in the safe compartment, because the safe compartment is associated with the absence of the shock. So it's like inhibitory stimulus that overtraining can acquire reinforcing properties.
So the animal, when they hear they have the cue and then they move the safe compartment.
Yes.
And then
I present a tone. Yes, I present a tone. Yeah. 5 seconds later, an electric foot shock. And if the animal associated both stimuli, once the tone is presented, he will run very rapidly to the safe compartment to avoid the shock. And then once he's in the safe compartment, he will develop relief and relaxation, and this has reinforcing properties. So and the animal is approaching to the safe compartment because it's reinforcing in the avoidant task.
And is there a signal to the animal so the animal will return to the unsafe compartment?
Sorry?
So after the animal moves to the safe compartment,
yes.
Is it another signal to the animal so the animal know it will move back to the unsafe compartment?
So there is, when the animal enters the safe compartment, a light is on, and it's on for 30 seconds or for 1 second. And when this time finishes, I use a box to take the animal and put again in the danger compartment for the next trial.
I see.
Sorry, I didn't explain that.
Okay, all right, thanks. Yes.
Okay, so there is a light that the animal can see. Okay, that is on during the period of the safe compartment. And when it finishes, I take the animal and I put it again in the danger compartment.
Okay. Carmen?
Yes.
I had a question. I don't know if you see the chat. So I see that
Actually,
no, that's okay. I see that the performance is worse in post thing. Is that the 10th day of post shift?
Yes. So the post is suppressive is when they are receiving 30 seconds. So this is the experimental group. Here he's receiving 30 seconds in safe compartment. Once the animal acquired the criterion, when the animal performed 5 consecutive avoidance responses, we change and we reduce time spent in safe compartment. It's like if I give the animal 12 pellets, I put 1 pellet.
Okay, yeah, my question was if it is typical to see the performance worsening in later post-shift sessions because the performance is worse in 10 than in 5.
Yes, it depends on the experiment. Sometimes we see the difference in 5, sometimes in 8, sometimes in 10. So it depends.
Okay, thank you.
Okay, so because it's also, I think that this response is like automatic. So the animal finally responds very rapidly. So maybe during the POSIT 5 criterion is still behaving in an automatic mode, you know? So maybe they need more training. Okay. Okay. So we have also conducted a number of studies with an appetitive instrumental SNC tasks. Here, animals are placed in a start box. So Marta did a lot of many experiments with this task, right? So animals are placed in the start box, and they have to run from the start to the goal box where they can find food. And what we manipulate here is the amount of food. So as in the previous experimental design, we again have a downshifted that receive 12 pellets 12 pellets in preset phase, and after this phase, they are downshifted to 2 pellets, and an unshifted controls that receive 2 pellets in both phases. So again, we compare performance during the posit phase for both groups. Here, the dependent variable is response latency. As you can see here, the results, this is the preset phase, this is the terminal performance. So here, we don't see differences, but it's because it's the final performance, but in the previous session, we see slower acquisition in the control group that is receiving lower amount of food, okay? And here, when we downshift animals and reduce the number of pellets, we see again an impaired performance. So here we see that the latency is higher for the experimental group, for the downshifted group compared with the controls, okay? So this is the instrumental successive negative contrast effect. And it's very interesting because in terms of the brain regions responsible, we know that there are some regions that are associated with this effect, but are not associated with other tasks, also involving frustrating non-reward. Okay? And this is our final task. That is the one that we are using now to analyze CFOS expression in the brain. And this is the task that I plan to present in SFN. This task involves giving animals sucrose concentration solutions, or solutions that vary in sucrose concentration. So again, we have a downshifted group that receives 32% concentration of sucrose in preset phase, and they are then downshifted to 4%. And again, we have unshifted groups that receive 4% during all training, okay? The animal doesn't have to show any anticipatory response. They don't have to learn anything. They just have to go and drink, okay? So the dependent variable is fluid intake. We can also use leaking frequency, time in contact with the zipper tooth, or just fluid intake. That is the dependent variable that we use in our laboratory. So here we again can see a typical result of consumatory contrast. This is the fluid intake in milliliters here. We can see that animals exposed to the 32 sucrose drink more during preset phase, but after the downshift, they show significant consumatory suppression compared with the control group. So both groups are receiving 4%, but consumatory behavior is much lower in this group because they have the expectancy of receiving something better. So again, this is a transient phenomenon. So after a number of trials, we see that both groups behave similarly. So this is also very interesting because in our present project, we won't see not only this initial suppression, but also the recovery, the patterns of recovery, because we know that they are individual differences. So some rats recover very fast, some other never recover, and some other, the majority, show this pattern, this curve of recovery. So we want to see if there are differences between females and males in this recovery, because that could be associated like a resilience or something like this, so the capacity of cope with frustration. Okay?
Hi, may I ask a question, Carmen?
Yes, of course.
Please go back one back slide. The view.
What?
Could you show the slides in the previous slide? Previous.
This?
Yeah. So my question is, can you control the weight of rats? Because, you know.
Yes.
Yeah.
Yes, because the fluid intake is expressed here only in terms of milliliters, but now we are using milliliters per kilogram.
Liter per kilogram. Okay.
Yes,
Yeah,
because now we have males and females, and males higher weight, and we are using milliliters per kilogram to control differences in weight.
Weight. Okay. Thanks. But apparently weight may affect the contrast, right? So even if I mean absolute weight may
Absolute weight?
Absolute means not the food intake divided by weight, but rather weight.
So what will be your suggestion?
I'm just curious about weight might affect the negative contrast. So
Yes,
so
it can affect.
big rats, you know.
So
Big,
yes, this is it's very interesting question, because our way to control the weight and how weight can affect intake, we calculate milliliters per kilogram. But maybe you have another better suggestion for us, or maybe you do it in a different way.
No, I'm just curious. So how
Okay.
might big rats may you know?
So we calculate according to the weight of a rat, what is the intake based on kilograms. Okay,
Yeah,
and
but
it's interesting because sometimes if we express the result only in milliliters, we don't see differences between female and males. But if we take into account the weight and we calculate milliliters divided by kilograms, then we see that females drink much more than males.
Okay.
So
Okay, yeah, thank you.
it is something that I want to present in SFN, maybe these sex differences.
Okay, thank you.
But if
Sorry, quick
you
question.
have a suggestion, if you think about a better way to control the weight, please let us know. Thank you.
So I had a quick question. Sorry, Carmen.
Yes.
Dr. Olga, what were you trying to say? Like rats that are bigger may have a greater propensity to take the sugar? Or were you like, how exactly is that what you were thinking?
Yeah,
The female, sorry, I
Sorry,
repeat, sorry.
I was asking if the reason why he feels that it may be a confound is because bigger rats may be more likely to take more sugar if they are if they're heavier because they've been taking more sugar. But
well, I don't
my
know.
thought, sorry, my thought is that or at least based on my experience, like dealing with rats, is that the weight would probably, like the frustration effect would probably be much greater than the effect of weight. So like the weight of the rat might introduce a little bit of variability, but in the end, all rats will react the same way. They're frustrated, so they're gonna take less than what they previously took. So either way, the average will go down, right?
Yes.
So that's one way in which that's and
So-
also them
So
having
what you mean is that a bigger rat can be more motivated to drink sucrose?
I think that's what he was thinking, that like
Yes, but in our case, it's the female that is smaller. The female is drinking more.
yeah, so I think what I was trying to highlight is that liking of the reward is not necessarily linked to like physiological appearance per se. It's very much like individual differences of like how do the rats value the reward.
Yes.
So
The hedonic value.
I think a lot of the hedonic versus homeostatic, right? So an animal might be bigger because they are homeostatically require more food, but they may not be like hedonic in that way, or like they may not prefer hedonic feeding, even though they are technically heavier.
Yes, but in terms of homeostatic value, we should expect more drinking in males than in females, but we see the opposite patterns. So, okay, so I will show you the result later on of females and males. So it's like tricky because we see this difference is impressive, but the contrast effect is similar for both. So I will show you, okay? Very soon. What time is it? Okay, so we know that the successive negative contrast effect can be manipulated by a number of experimental manipulations. Some of them reduce the effect, and some other enhance the effects. And we also know that the experiences with reward evaluation can enhance or reduce behavioral, many different neurobiological correlates. So I'm not going to explain this in detail, of course not. I just want to show you some of our contribution, the contribution of our group during these years to this topic, okay? So the successive negative contrast effect has been extensively studied. So in terms of pharmacological drugs, behavioral treatment, brain regions responsible for the effects. So this is some of the things we have done. For example, we have injected GABAergic anxiolytic in the aversive task, and we found that this task responds very similarly to the consumatory task, so it has predicted validity. Marta, another graduate student, conducted an experimental series with a strain of rat selected for high level of anxiety. We found very consistently that this strain, the Roman low avoidant rats, show a higher consumatory successive negative contrast effects, not only consumatory, but also instrumental and aversive effects. So there are like a genetic influence of this effect. But we also find that some environmental treatment can also increase or decrease the size of the effect. For example, partial reinforcement is a treatment that can induce counter conditioning and can reduce resistance to subsequent reward loss experiences. So I don't want to talk very much. If you have any specific question, just let me know. Okay. There are also a number of experiments that have explored the neurobiological correlates of this effect. In our laboratory, and Marta Sabariego did it, we analyzed differential gene expression associated with successive instrumental successive negative contrast. We compare low and high avoidance rats in the hippocampus after experiencing reward downshift, and we identify a number of genes that were differentially expressed. We also conducted an experiment in Austin, in the University of Austin, in which we analyzed the changes in the level of PCREF, this marker of neural plasticity, and we found differences in the anterior cingulate cortex, dorsomedial striatum, amygdala, I think, and infralimbic cortex, I think. And this is the last one at the new, that is the study of the brain correlates of the successive negative contrast effect using CFOS brain expression analysis. Okay, so this is what I want to present in SFN. So this is based on this picture that is from your special issue, Marta. So as all of you know, frustrating non-rewardable loss are included in the negative balance system of the RDoC domain criteria approach. We have a lack of knowledge about some topics, for example, the existence of sex differences. And with respect to the contrast effect, there are not systematic studies comparing females and males in terms of how they react to the reward downshift and how they recover from the experience. So one of the goals of our project is comparing female and males. And also the other very important goal is, these are the goals, sorry, is identifying the brain regions responsible for the consumatory effect. So we want to analyze to conduct CFOS expression quantification across a number of regions of interest. Okay, we want also to correlate behavior and CFOS expression with the final goal of develop a functional connectome based on CFOS expression patterns across POSIT session and applying or using graph theory analysis. So our goal is develop a functional connectome for male, a functional connectome for females, compare both, but we are having problems now. Try also to analyze how this connectome changes in terms of functionality, depending of the recovery pattern. Okay? So this is our model, neurobiological model. This model was proposed in 17, sorry. So these are the brain areas that had been shown to be related with the consumatory successive negative contrast effects. So we know for sure that all of these areas are related with some of the components of the consumatory task, including the reward processing, reward comparison, the emotion, the frustration, negative emotion or frustration. So according to this model, we have a number of areas that have been analyzed in isolation. So they never put together and try to get how they are connected and how their activity can influence each other. So the goal of the project is try to develop this connectome and put together and compare pattern of CFOS expression across all of these regions. Okay. So this is the general process. It's very easy. It's much easier than it seems. It's just that animal will be exposed to 10 days with access to cherry 2 sucrose. Okay, this is the experimental group. Of course, we have the control groups. And on session 11, they will be downshifted to 2% sucrose. Okay, we downshifted from 32 to 2% sucrose. And 90 minutes after, they were sacrificed in order to proceeding with the immunohistochemistry for CFOS. Okay, so some animals were sacrificed after session 11, some other after session 12, after session 13, and after session 14. Because as I told you before, we want to analyze not only the CFOS expression related with the initial downshift, with the initial consumatory suppression, but also with the subsequent recovery. Okay? This is more or less the steps that we want to do. So after the consumatory task, animal will be sacrificed, we will slice, we will proceed with the immunohistochemistry, we will analyze, we will quantify level of CFOS expression in this region of interest, and then we will try to use the graph theoretical analysis to identify patterns of correlations between regions and try to analyze the properties of the connectome, the property of the network, okay, and how the network changes during the POSIT sessions, okay? So these are our preliminary results. This is the behavior. This week complete like 4 replication. We have almost 200 brains now. Okay, this is the behavior. These are the animals that were sacrificed on session 11, session 12, session 13, and session 14 as something that is very consistent here. This is the dependent variable, the sucrose intake, is expressed in milliliters per kilogram. Okay, here is not milliliters, it's milliliters per kilogram. And here what you can see is that female animals drink more sucrose than males, but especially the highest sucrose concentration. So it's very consistent across the 4 replications. So here these are females receiving 32. These are the females, these are the males. Okay, so female drink more. And these are the groups, female and male receiving 2%. Here you don't see any difference. So there is an interaction between sex and sucrose concentration. But in some other replication, we see that for the 2% concentration, by the end, later on in training, we start to see differences. Can you see this one? On session 11, 12, and 13. And also here. Here we see that, again, female differentiate and drink more. But we need more trials to see these differences. With respect to the 32% concentration, the differences are present in the second session, okay? And this is a very consistent pattern, okay? Something that we saw in an anecdotal way is that the animal not only show sucrose or consumatory suppression, but also start to show different response when they are exposed to the downshifted event. So this is an animal, impressive phase. At the animal, the only things they do is drinking and drinking and drinking for 5 minutes, okay? But this is what can happen. This is the positive session 11. This is one of the animal. They are trying to escape. So we are watching now, we are now recording these behaviors in a more systematic way. Because, for example, this is biting. This rat is biting the super tooth. Some of them, there is an increase in ambulation. Some of them reject the solution and they go to the other section of the box. So this is something that we saw anecdotally, but it's very idiosyncratic because, for example, this animal that tends to jump, he tends to jump every single positive day, okay? So it's very consistent, and I think that we can detect individual differences and different ways to cope and to show, to express frustration. So this is something that are doing right now. So this is an experiment that are conducting. Today is day 10. Tomorrow is the first positive session. So we are recording all the session now, and we are going to analyze this. We are going to do this ethological analysis. We are to include ambulation time, reading, head dipping, jumping, other escape-like behavior because they try to like climbing. ¿Se dice climbing? No? Climbing. Contact, burying, because some of them also do this kind. So we expect to find differences, and maybe we expect to find differences between females and males. Because in terms of the consumatory suppression, we don't see differences. So the consumatory contrast is similar for both females and males, okay? The initial suppression and also the recovery. But maybe if we analyze these behaviors, we can find differences. For example, I think that, I don't know, because I have to measure this systematically, I think that females have higher ambulation. They have more exploratory behavior. They are much more active in the box. So I don't know what's going to happen tomorrow. Okay.
Sorry, quick comment, Dr. Torres. I find this so interesting because we're kind of looking at similar things now. I don't know if you remember like Dr. Green and I's work at all, but like we measure duration of pressing in an operant box,
Yes.
as well as force of pressing. And the way I'm thinking about those is as emotional reactivity. So they're displacing that frustration into how they interact with the levers.
Yes.
So I wouldn't be surprised if you found a way to measure force on
Yes.
the tip of what's that called? I don't know what it's called.
The zipper tube. Yes, we cannot do it because we don't have the device. But I can tell you for sure that the way they interact with the zipper tube is completely different. So if you see here, this rat have two zipper tubes. One has sugar and the other one has water, because now in this experiment, we want to analyze whether or not the sucrose,
There's like
the
a
down-shifted sucrose develop aversive properties. So this will be like a preference test. But during the preset session, what we see every single day that when the animal is interacting with the water zipper tooth, the way it interacts is completely different. Sometimes they bite.
Yeah.
They bite when they realize, when they notice that this is not sugar, that is water. So I think that tomorrow we are going to see this kind of behavior. So I don't have the opportunity of measuring the force, the pressure, but I think that visually we can see a different pattern of interaction.
Yeah, for sure, for sure.
Okay. And these are some of the data we have from the CFOS analysis. Okay, these are some of the regions that we have already analyzed. This analysis is complete, I think, but we are adding. So this is the we have like a very big file, Excel file, in which we are adding every single information we have about CFOS and different regions. So this is an experiment, an study in which we measure CFOS expression in the hippocampus, and according to previous literature, we didn't find differences because the consumatory task is not related with the hippocampus. It's hippocampus independent. Okay, so according to this data, we didn't find differences between downshifted and shifted in the hippocampus in terms of the hippocampus CFOS expression. Okay, but this is the avenula. And in the lateral avenula, and especially in the medial portion of the lateral avenula, we found increased CFOS expression in the downshifted groups. So we know that the lateral avenula is involved in the detection of reward omission, and we have this paper now in preparation. We are writing now the paper. I think that this is more or less complete. This is in process now. So we improve. So the most important challenge or problem we had is trying to setting up the CFOS procedure to be sure that all the concentrations, the antibodies, that everything was okay. So we had to do many minor adjustments. So at the beginning, we had problems to get good picture, good images, good signal. But now I think the technique improves a lot. Marta Valero is working very hard with that now. And we also have a microscope with a scanner at the University of Granada, where we can scan, I don't know how many slides. Marta Sabariego, this is amazing. Now we can go to Granada every week, and we can scan one whole brain in one day. Okay, because we have a scan that can process all the slices very, very fast. And the quality of the picture is amazing. So this is some of the picture. So thank you, Alejandro. Thank you, Marta. Thank you, Tony. Thank you, everyone who helped me to present this slide today. These are preliminary data in the amygdala, the basolateral and also the central amygdala, the shell, and the core of the nucleus accumbens, and different portion of the insular cortex. This is still preliminary, so we only have a few number of brains, so we have to extend and we have to replicate this data. Okay, and this is what is coming soon, because now some students that are working with Tony are analyzing the DTA, locus coeruleus, the raphe nuclei, because we know that the raphe is connected with the lateral avenula. So we think that maybe we can also find differences in this portion of the brain, also in the different division of the orbitofrontal cortex. So this is what is coming this year. So I hopefully expect to have like a preliminary network or connectome after the summer. Okay, so the goal is using this region of interest, this model, to develop, and this is where Antonio Ibanez can comment something if he wants. So This is not ours, so this is like something similar to what we want to do. So we will have a network containing nodes. Every node is a region of interest, and the black lines are the anatomical connections. So we are using the GMT database. I don't know whether if you know. So it's a document, it's an Excel file where you can find all the connection between among all the brain regions. So we can build a connecton based on this anatomical information based on the functional connection. So we plan to have like a matrix, a matrix of correlations among these regions, based on the strength of the connections. We will develop this connecton, okay, and we'll analyze their properties. So we can analyze, for example, small world properties, reciprocity. So there are a number of properties that can be analyzed using the graph theoretical analysis. I'm not an expert at all, so I have the collaboration of Antonio Ibanez, who is the person who will help us. Now we need to add more data because we still don't have enough data to start with this analysis. But we are proceeding really fast now, I hope. So I hope to have nice data in the near future. So that's all what I wanted to show you today. So maybe it was too long, I'm sorry. No, it's 1 hour, more or less. So I hope you understand my English, sorry.
That was great. Thank you, Carmen. We have a couple of minutes, well, maybe a minute, for questions or comments.
So your kid is your research even interesting because of this, the lever pressing duration is pretty similar to what we see when they are trying to bite.
Exactly. What's
Yeah.
interesting is with the force lever, and I think we mentioned this last meeting, the force lever, it's not a press, you know, it's a bar. So they interact with the bar however they want. And so a lot of them will bite, some of them will like do a mixture of pressing and biting. So now we're also going to start recording for those individual differences of like, okay, during the sessions that they're frustrated, how does that shift their normal like behavior with the lever? It's very interesting. I'm
I
excited.
think you will find a change in the pattern of interaction.
For sure. I mean, I
so
see it. Like you said, you see them behave differently, literally.
Yeah, of course.
And we've even thought, I mean, we've flaunted this idea for a long time of like giving them an option to escape the box.
Yes,
when it's frustrating, like ending the session or something.
Yes.
Because they just be like, oh my God.
So there are some experiments. So
Huh?
there is some experiment made by Amsel with an alley. So if they have the opportunity to press something or do something and escape,
Right.
so the alley is open, they will do it and they will learn to escape from frustration.
Yeah.
Yes.
I thought that idea of having the water was very interesting also because of that, because in a way it's an escape, right? It's like, okay, I don't have to be drinking this if I hate it. And you can see like maybe that switch with the water after
yes,
the
this is what we expect to see tomorrow. So this switch. So they will try. So this is I don't like this sugar, so I want to try the water, but I don't like the water. But I think because now we are calculating the preference ratio, and the preference ratio is 1 for all the rats, 32 and 2.
So you will
they
see
don't
something,
drink water at all. So I think that tomorrow they will shift.
There will be some,
and they will increase. So they will still prefer sucrose, I think. But I hope that the preference goes down.
Yeah, also to check whether there is something else there, right? Like that flexibility to change behavior and check whether there is another source of reinforcement.
So
Is
we
this
are not,
something?
yes, we are not only to measure the fluid intake, because maybe they interact with the super tube, but they don't drink. So we are also going to record time. For example, we are going to divide the box, and we are going to record how time they spend in the section where the sucrose super tube is located, or
I see.
how long they stay in the other. Okay.
Has this been done before, this procedure of having the water choice?
No.
Or is this
No, this is new
really
because
cool.
this is part of the grant. So one goal was analyzing sex differences in the differential components. So one component of the consumatory contrast is aversion. So we don't know whether or not the down-shifted sucrose becomes aversive or just less hedonic.
I see.
So we want to see
because
if it's aversive, the animal will prefer the water.
The water, yeah.
But
really cool.
It's tricky, it's difficult to see because we cannot deprive animal from water. Because if we deprive, you don't get the contrast effect because they drink and drink. So if they are not deprived from water, this is something that we were discussing a lot. So we were not sure what to do. So we don't know. There is another way to analyze whether or not the down-shifted sucrose is aversive, is analyzing the microstructure of the leaking behavior, because the way they leak is different if the solution is aversive or is appetitive. But for that, we need a special device. So we need a collaboration with people from with Mauricio or with Ricardo Bellón in Madrid, because they have the device and they can not only analyze, because they used to drink in burst. So they drink, stop, drink, stop, drink, stop. So you can analyze the pattern, the microstructure. So the interval between one burst and the other one. There is extensive evidence showing that when solution in aversive, the pattern of leaking is different. So this is something that we want to do in the maybe after the summer.
Okay.
The microstructure, and also we want to analyze the facial expression. We want to record the face of the rat, because they also can show expression of aversion, this gape behavior of hedonic value. So we can also compare. So it's like an experimental series. So I hope we can do these 3 experiments.
Really cool. Awesome. Okay, so next time we will hear from Jan and Ellen. So we are looking forward to it, and I will send.
What is the date for the next one?
It will be April 15th, but I will send a calendar invite like I did today. Okay, so that everyone has it in their calendar. And maybe we have more time on April 15th, we can discuss some of the proposals that, for example, Mauricio proposed that we have a website. Maybe we can talk about that in April, because we don't have time today, but yeah. And if anyone has any more feedback or suggestions for Carmen, feel free to email her.
Yes.
Carmen, can you put your email in the chat?
Yes.
Or we have the email. It's in the chain, in the email chain.
Have it?
Yeah, we all have your email. So if anyone has any suggestions, feel free to email Carmen, and otherwise we'll see you in April.
Okay. Thank you very much.
I'll send the recording once it's processed.
And I will forward the link also to Antonio, Tony,